Okay, I think we can go. Go ahead.
My name is Megan Ricciardi, and my project focused on pantethine-based inhibitors of the quorum sensing enzyme set-by. So what is quorum sensing? Quorum sensing is a form of bacterial communication that uses chemical signals called autoinducers, which will be referred to as AI. This form of communication is used to monitor cell density and initiate group behaviors. The responses from quorum sensing include bioluminescence, biofilms, and pathogenesis. This is a figure that shows both of the receptors used, both of the enzymes used within the quorum sensing mechanism. To start with, we start with the AI synthase that produces this AI autoinducer molecule that is shown here in the yellow spot. These molecules are very tiny and can diffuse in and out of the cellular membrane as shown here. And this is a figure of what some of these autoinducers tend to look like. When there's ample autoinducer available, it can then diffuse back into the cell membrane and interact with this AI receptor as shown here. This AI receptor then interacts with the DNA and can then transcribe quorum sensing related genes. As I said before, there are many behaviors controlled by quorum sensing. Agrobacterium tumefaciens controls pathogenesis, and that is found in plants. Pseudomonas aeruginosa controls biofilm formation and virulence, and that is found to be a human pathogen. Chromobacterium violation controls violation production, and Vibrio fischeri can control bioluminescence. As I said before, we have been focusing mainly on Pseudomonas aeruginosa because it can create biofilm formation and virulence within humans. So the overall goal of my project is to create an chemical inhibitor that'll inhibit this AI synthase shown over here and ultimately stop the quorum sensing pathway and the transcription of these target genes. So to start off, I'm going to talk about the different parts of the AI synthase. So the AI synthase has two separate binding pockets that meet in the middle and use these two reagents right here in order to form the autoinducer. So in one pocket, acyl ACP binds, and in the other pocket, SAM binds. When this happens, there then becomes an acyl transfer as shown here, followed by a lactonization to form the autoinducer shown over here. Previously in the lab, we have made a molecule called JKR, made by my colleague Julian Kellner-Rogers, that mimics this transition state with the acyl donor on it. And this has been proven to be an inhibitor of this AI synthase. It binds within this SAM pocket of the acyl ACP. What I want to do is make a mimic that'll bind within this acyl ACP pocket of the AI synthase. As I said before, I was creating inhibitors to target the acyl ACP binding site. This right here is a crystal structure of BJI that has the acyl ACP bound into the pocket. This pocket is, as I said, for acyl ACP, and then this one over here is for SAM, and in the middle, they meet in the back here. This right here is the native substrate for acyl ACP. As you can see, there's an ACP protein held on by a phosphate group connected to this pantethine-based arm. So what we would like to do is create pantethine-based inhibitors that look similar to the substrate minus the protein and the phosphate. And within this green box, I will be changing up the acyl tail groups that are connected. Right here is shown as a thioester, and I would like to create thioethers as well as amides. As seen here, this is again the native substrate. This is the acyl tail, and this is the pantethine-based portion. My inhibitors will include thioethers shown here, connected to various tail lengths, a C8, a C10, and a phenethyl thioether shown down here. And then I also created inhibitors that have amide-based ends. So this is right here an amide, and then the sulfonamide connected to the tails. This is the overall scheme of how the reactions go through. It's pretty big and bulky, but as you can see here in the colored boxes is the final product. And then over here, you can see that they all start from a similar starting point. So I will now go into a couple more in-depth steps. We'll start with a thiol-based synthesis that makes this C8 thioether, as well as the C10 thioether and the phenethyl thioether I mentioned before. So this starts with an acetal formation. So we start with pantothenic acid to create then this acetal and carboxylic acid. We then add on this sulforamine with carbonyl diimidazole in order to form the thiol end group. We then do an SN2 reaction with this BRC8 in order to add on the carbon tail to the thiol and make the thioether. And then we reduce the acetal with the 1:1 formic acid water to get the final product. For the amide synthesis, this was the same exact route that I used for the amide and the sulfonamide. Again, it starts with pantothenic acid getting protected with this acetal group and carboxylic acid. That then gets a diamine added with a protecting group on one end, and again, carbonyl diimidazole, and that then adds this diamine to the carboxylic acid. We then reduce the protecting group on the end to make just an amine. And then we use carboxylic acid chloride with a C8 tail shown here in order to add that onto the amine and make the product with a protected acetal group. And then we reduce the acetal again with 1:1 formic acid and water to get the final product. Then these molecules are put into an autoinducer synthase enzymatic assay. This takes advantage of the fact that this ACP thiol is produced when normal autoinducer is available, and uses this reagent called DCPIP that starts off blue and then goes to colorless when this ACP thiol is around. This chart here shows how normal enzyme works as shown here. This is when the enzyme is functioning totally properly. And these flatter lines is when the enzyme is when there is either no enzyme or the enzyme is heat destroyed and no longer functions. So as you can see, there's no color change at all. And then down here, it goes mainly to colorless. So for me, I tested my molecules in the same exact enzymatic assay. To start with, I tested my C8 thioether, my amide, and my phenethyl thioether. So within this assay, on the y-axis, we have the rate, and on the x-axis, I showed the various different components. So with no enzyme, there's obviously no enzymatic rate. With SEP-I alone, without any inhibitors and just SEP-I working on its own, it has a very strong rate. When JKR is added, the other known inhibitor, we add it and it reduces the rate pretty significantly. And then when we test my 3 inhibitors, there's no significant change in the rate, indicating that they have no inhibition properties of the SEP-I enzyme. I then followed this same procedure with my sulfonamide and my C10 thioether. Again, we use the same enzymatic assay. with the rate shown here. With no enzyme, there was no real rate shown. With SEP-I alone and working properly, there was a strong rate, enzymatic rate. With JKR, there was a major decrease in the enzymatic rate showing inhibition. And then with my two molecules here, the sulfonamide and the C10 thioether, there was again no significant decrease in the rate. So this led to the conclusion that my first design did not work so far and that the compounds I generated did not inhibit SEP-I. So then this created the need to create another design. So upon further investigation of the enzyme, we found that there was an innocent cysteine buried deep within the acyl group binding pocket that could be targeted with various electrophiles. So this is the scheme showing the mechanism of how this cysteine can be used with the electrophiles. The sulfur part of the cysteine can then attack the electrophile and hopefully make a covalent bond in the back of this pocket, creating an inhibitor. This is a crystal structure of TOF-I bound to another known inhibitor called JAC8. And this arrow points to the cysteine that is found at the back of this pocket. As you can see, the inhibitor and the cysteine are fairly close together, leading to the conclusion that this interaction may occur, creating the covalent bond. I then created, designed two different molecules to be synthesized. I synthesized this C7 electrophile right here with the bromine, and I was in the works of synthesizing this isothiocyanate to be tested as well. Unfortunately, I didn't get to test the bromine, but that will hopefully happen soon and create results of a covalent bond being formed. Future directions include testing this bromine electrophilic inhibitor, as well as synthesizing and testing the isothiocyanate electrophilic inhibitor, as well as design and synthesize further electrophiles to be tested against the SEP-I enzyme for hopefully covalent inhibition to occur. I'd like to offer special thanks to the Ulrich Lab for taking me in for the past 3 years. It's been a lot of fun, and I've definitely learned a lot that'll be useful in my future. And I'd also like to thank the Ithaca College Chemistry Department for, again, taking care of me in my 4 years at Ithaca College and allowing me to get to this point in my career and move forward into the future. Thank you.